We're trying to learn a new language, the language of the plant chemistry or the insect chemistry. We try to decode each words and try to understand what the sentences they try to put out there.
Hi, I'm Laura Riley, and this is Research Matters, a show about the Cornell researchers who are finding solutions for the world's biggest problems. What if the key to understanding the health of our bodies, our environment, and the crops that feed us was floating all around us? What if the invisible world of scent, those chemicals whispering in the air, held clues waiting to be decoded? Today we're diving into that world with Christophe Duplais, Associate Professor in the Department of Entomology at Cornell Agritech. His work asks a surprisingly bold question. Can we collect and analyze smells to understand the world around us? Welcome, Christophe.
Hello. Hi.
So I would love to start with kind of a big idea. Most of us think of scents as kind of, this personal, like, ooh, that's a lovely cologne, or, who burnt the toast, that kind of thing. But your research treats smell as kind of an information system, almost like a language. What does it mean to read smells in nature?
So reading smell in nature means it's really about trying to understand the words that help us to communicate, right? When we communicate, we use words to make sentences, but the smells each molecule can be associated with a word, something saying. And so if you put different chemicals together, you can start to have a sentence. And so I think that since the beginning of the evolution, any organism has been trying to interact with each other by communicating, whether using smell or other ways. But the smell is a very easy one to communicate.
So who is signaling whom? Like, give us like the array. I mean, we hear about pheromones. It's a very mysterious kind of, thing in humans. But who are the signalers and who are they signaling?
So I think that I will say that there is a topic called multi-trophic interaction, where you go from the plant to the herbivore, to the parasitoids of the herbivores. So it's like the good way of starting this conversation, because when an herbivore is showing on the plant going to start to release a bunch of molecules. Some of the molecules, they have no function at all. But some of them, they actually are trigger for parasitoid who's going to try to... The parasitoid is going to figure it out that some plant is being attacked by a herbivore, that the parasitoid can put lay eggs inside. So that's the multi-trophic.
So the plant is deterring their predator by attracting a predator for that animal.
The plant is calling for help for some organisms that can help the plant to defend against the attacker. So the question is where the selection has been made here. Is it the olfactory receptor of the parasitoid, or is it the chemicals being released for the plant who's being the selection pressure? This is very hard when you've got an emitter and a receiver to see the dynamic of evolution of who's starting it first, the communication.
Are you developing ways of kind of eavesdropping on the plants that are sending out these messages?
Exactly. We're trying to learn a new language, the language of the plant chemistry or the insect chemistry. We try to decode each words and try to understand what the sentences they try to put out there.
Okay, so this is sounding very kind of almost psychedelic right now. So, but how do you, but how do you collect the scents? What does that look like in the field?
So in the field, it's very hard. And most of the time, there is this technique that I've been using for a very long time. We use absorbent. So we use kind of dispenser that absorb the smell. So sometimes when you try to filter your water, use charcoal, right? Mm-hmm. But actually charcoal is a very good absorbent for lots of volatile. So even in environmental studies, when we try to figure out toxins in the air, we use this kind of absorbent like charcoal who trap the molecule, and then we can dissolve them either by using absorbent or by just heating. So you release the gas that's being absorbed into this dispenser. But it's still very biases. There's lots of biases when we collect volatile. And so some of the there is one instrument that we can bring in the field, but it's a very expensive instrument. And when I try to buy this instrument, which costs like $500,000, when I ask if I can see one in New York State, they say like the last time someone buy it in New York State, actually he was struck by the lightning.
Oh.
Because you put a piece of metal and electronics in the middle of the field, and it's so you get burnt. But I feel like now we've got new technique that has been driven by the food science, the food industry, and we've got like very, very tiny piece of plastic that we can drop around, and they absorb very efficiently. And
that sounds way less expensive than that $500,000.
Very less expensive. But it's still no real-time monitoring. So we're still to bring back the sample in the lab and analyzing it. So it's crazy that with 2025, we can have probes everywhere on Earth. It's still very hard to have the detection of smell in real time in any place.
Is that an indicator that smell is something we haven't taken as seriously as the other senses?
We haven't taken seriously. I think it's a very one to break through and to decode carefully. Because I think that we know that humans have smells. Pheromones, we haven't figured it out. And we have
I thought that was like what Axe body spray was for. Or I thought that's how we kind of attract each other, is this pheromone.
There are a few experiments that test how behavior for pheromones, and we know we have behavior for pheromones. We haven't figured out the chemicals behind that.
Okay, so for humans, a smell, goes from the nose to the olfactory bulb to the amygdala and the hippocampus, I guess. Is that for all vertebrates? Do all vertebrates smell the same way?
So I don't even think that every human smells the same way. I think there is lots of variation even for us. So then across different species, definitely something that smells. I can think about the crops flower. So you got this plant who smell like crops to attract pollinator. For us, it's going to be the smell of the crops. So it's going to be deterrent. But from some of pollinator, it sure smells like heaven for them because it's an attractant. So I think it goes beyond the type of brain decoding function we have. It's about what is these smells make, how this may change our behavior, and what's the driving force of that.
So are we all, across invertebrates, are we all driven by smell?
We are all driven by many cues. Smell is one of them. But I'm not quite sure that, we are the only species who, I'm not quite sure about that. We are the only species who use smell for perfume, right? But are there other species who take benefit of the smell to camouflage or to even like do courtship? I think this is not belonging only to humans. I think lots of some other species do that.
Yeah, makes sense. All right, let's talk a little bit about agriculture and kind of the application of this. So, we've been through, 80 years of very chemistry intensive agriculture, and it's gotten us in a little bit of a pickle in some ways, depending on who you talk to. Large scale commodity row crop, pesticide intensive. Are there ways that your research can bring a different kind of paradigm to how pests are dealt with in the field?
So I'm always trying to make this analogy between personal medicine and precision medicine, and personal agriculture and precision agriculture, because in medicine, we try to detect the disease before it spread out, or even if the first cancer cell appear, we try to detect that. It's the same challenge in agriculture. Every time we've got a new disease, we want to make sure that we can detect it before it spread all around. And so smell is one of the good things to track down, because when we are sick, we smell differently. Whether any organism with some kind of disease will have some cues going to be different when they are not affected. So figure it out when one single plant is getting infected by any disease, help us to tackle down this issue and spread less pesticide, because instead of spraying the whole field, if we can localize precisely where the infection is, then we reduce the chemicals we put in the field.
Well, I know that dogs have been used. Obviously, we have a long history of pigs being used to hunt truffles, but we've also had dogs that in recent years have hunted things like in Florida. There's a boring beetle in avocados that it's like drones have identified parts of fields that have like canopy damage, and then dogs have been sent out to scent essentially where the disease is or where these boring beetles are. Is that an asset, or is that a strategy that could be used more broadly?
It is, but the dog training has limitation. I can tell you an example of the leaf whorl virus, which is a virus that attacks grapevine, and it changed the taste of grapes. So they have zero tolerance in California to have this virus. They have to remove the whole vine.
Wow.
Why now we have really big trouble detecting that because we have to use PCR tests, and we train dogs right now to detect the vine. They can smell when the grapevine has this virus, but they still have limitations. So I'm working with some different companies right now who can develop new type of detector that can provide real testing on site. And we try to train the device right now using AI to differentiate the smell of the plant when they have the virus or not.
So some kind of, this isn't dogs doing this. This is a device that detects the scent.
We try to have the same sensitivity as a dog nose, basically.
Wow. All right. So the goal is to detect these diseases before they become cataclysmic for a particular crop or widespread. Are there ways of introducing smells as a protectant for crops? And what would that look like?
So absolutely, because sometimes you just try to put the pest outside your field. And so there is like this very actually not very recent. We've been using that in Africa for a long time. It's called the push-pull system. So you put in the middle of a crop, some plant gonna be a deterrent, and you put outside of the crop of the field some attractant plant. So you got this push-pull system that push the pest out of the field, attract them outside the field. So this push-pull system has been used, and we keep using it because it's a very good alternative to pesticide. Absolutely.
So it's kind of like, I've always been told to plant marigolds at the edge of your garden to keep certain pests away.
Yes.
So it's that kind of.
Exactly.
So the pull part, are there ways of, I know that in a lot of biodynamic gardening or farming situations, they're trying to attract good predators for the bad bugs. Like, are there things that attract these? I don't know whether it's ladybugs or whatever.
Yes, and that's the difficult part of this challenge of which molecule attract which insect or which beneficials, whether you're microbes or nematodes, and how much eavesdropping you're going to have from species you don't want, who's going to adapt to the flavor you put in the field.
Wow. Yeah. I can see.
Being selective and make sure that this molecule attract only one species or just the good ones is very, very hard.
Yeah, the adaptation possibility, the learning of these animals. Sure. Well, so I'd love to talk a little bit about kind of human health, and are there ways of using these kinds of strategies in diagnostics for human illness or in thwarting human illness? Like how could smell be brought to bear in that situation? So
when I was based in French Guiana, I remember like a skin doctor telling me that he can recognize lots of skin disease because of just the smell the patient's going to have when they just enter into his office. And he knew that this one's got leishmania, this one is malaria. And even the smell of each disease. Again, the challenge is some of the disease going to have the same smell, going to produce the same smell. And so in this case, it's kind of like, it's kind of hard to tease apart which one's going to be. So we're not going to be very selective, but some disease is going to be very selective of the smell. Why now? I know that some different companies are trying to get diagnostic for COVID-19, and they've been very successful in UPenn, for example. They design a sensor that can, using the smell of the blood, or the smell of urine, and now they got the smell of the skin, they can discriminate if you got COVID-19 or not. And so now the big battle are, for example, cancer, that when we detect them, it's already a stage 4, stage 5. So ovarian cancer, prostate cancer.
Yeah.
When we detect them, it's already too late. So having proactively be able to have early detection of them will be a breakthrough in medicine for sure.
So I think there was a piece in the New Yorker maybe last year about a woman who detected Parkinson's, I believe, by smell. And I am assuming this is just an anomalous, like freaky sense of smell. Are there people who, like a super taster kind of thing, who have unique abilities to perceive things like that?
Absolutely. I think it's like the absolute ear, right?
Mm-hmm.
Some musician can detect any notes. Some people have gustatory receptor that are like way beyond the else. So there is some superpower humans out there. Sometimes I'm not quite sure they're aware of their ability. And now the question is like, now that they are aware of the ability, what they can do with them. And I think this person who can smell Parkinson, I would love, to understand how she decode that and how she can figure it out which smell is associated with the disease itself.
Yeah. All right. Well, so in terms of next steps for you in your work, what are the big questions that you're asking right now?
So it's funny because this morning I was actually with my good colleague Scott McCart and Danny Dyer working on the small hive beetles. So we got lots of problem with honeybees. And there's lots of pests. One of them is actually small hive beetles that infest the hives. Mm-hmm. And we just try to ask this question, are
these varroa mites or
they are not varroa mites.
Okay.
They are small beetles. Mm-hmm. And they hit the brood, they eat lots of things. And when you get the infestation, it's really, really bad. And why now? We are not quite sure the prevalence of these pests in North America. We know they're here for sure, and it's a big problem in the south, not so much in the north, but it's coming up. Now the question is like a simple question: how they detect the hive? Mm-hmm. What the smell of a hive? Do they prefer the hive when it's infested or not? Are they preferring honeybees, or are they going to go for bumblebees? So we try to figure it out. What the queue they're looking for, whether it's a long distance queue or a short distance queue, to see if we can do better trapping system, because right now we don't know how to deal with them.
Can you run interference? Can you introduce olfactory kind of bait and switches that make these pests go somewhere else?
Exactly. Like try to make sure that the trap is very close. And so they think they go to the hive and they go to somebody else to send a different trap.
Yeah. Oh my gosh, that's fascinating. All right, well, so obviously in agriculture, the big money is on there are 5 crops that let's just call it corn and soy primarily, and then you have wheat and rice, and I guess cotton is probably the number 5 or whatever.
For the field crops.
Yeah, for field crops. So, I mean, those are corn I think is something that has a number of predators and pests that are problematic pretty consistently. Are there any ideas there that could be brought, could be erased or eradicated by an
absolutely.
approach like this?
So one thing we try to deal with right now when we talk about precision agriculture is making sure that we know when the pest or the disease is right here, right? And to do that, for insect pests, we track them down by using sex pheromone. So we got traps. We put the sex pheromones of the female that attract the male. So we trap the male, and we know exactly when the population to build up. And then when there is a threshold, when we know that the population is too high, we have to spray. So this timing is very important, because some of the crops for fresh market for corn, for example, they can spray only one time this insecticide. So they really have to be very careful of when to spray exactly. They want to have the perfect timing. And to do that, we have to make sure that we can trap the insects based on the sex pheromone very accurately. And we do that not for corn. For apple, it's the same thing. With scaling moth, with lots of crops, we have to track down the insect to make sure that we can monitor the dynamics of either it's an insect that is overwintering here, or whether it's an insect that's migrating from the south.
Mm-hmm. So I know that Cornell has kind of a long history in this kind of research in Geneva. Can you talk a little bit about kind of your predecessors in the Cornell environment?
So I will say like the big names that we know at Cornell are Jerry Meinval and Thomas Reisner. One was an entomologist, one was a chemist. They were friends because they were playing music together. And together they are one of the founder of the chemical ecology, the field of chemical ecology. But there's so many other names. We got lots of new generation that comes right after. Meber and Malm was one of the first PhD students who cracked down the coevolution of toxins and adaptation in plant insects. She's a member of Academy of Science. So we got the generation after generation of chemical ecology in Cornell, which is amazing. But for agriculture specifically, I can talk about someone that is also a major figure that not everybody knows about because he was more in the field of agriculture, which is Wendell Wolof. He was a chemist like me, who was like doing organic chemistry and shift completely to agricultural chemistry. One thing that he created back then in the 70s, which is like outstanding, because of his skills of analytical chemistry and all the entomology physiological part we have, he created something that can help detect what the insects smell. So he created a device where he can mount the antenna to electrical wire, and when we put smell in front of this antenna, there is a signaling going through the antenna. So we can recall the signal, the electrical signal, and we can figure it out which molecules are detectable by the antenna. Mm-hmm. By doing that, he was able to detect over 70 insect pest pheromones that are still being used right now in the field to detect insect pest dynamics of migration.
Wow. So it sounds like the idea that organisms communicate chemically has been around for a long time and kind of have been building towards this, defenses and lures, essentially, the stick or the carrot, I guess. So what are the next logical questions in this field?
So I'm very interested in chemical ecology, but I'm also very interested in chemical evolution. So understanding like the evolution of the chemistry, the evolution of these pheromones, what are the constraints, the evolution of the receptors, what come first? And can we track down how many these olfactory receptor and molecule combination are the same? Do you always use the same receptor to detect the same smell? All this evolutionary trade-off and constraint are very interesting to have a broader understanding of how we can decode better and faster any smell.
Christophe, before we go, if our listenership today has a real interest in this topic and wants to learn more about our sense of smell, do you have any book recommendations?
Yes, absolutely. I got an amazing book that is not only about smell, but it's about all the cues in the world by any organism. And this book is by Ed Young. The name of the book is An Immense World, and is really put yourself in the skin of an organism and how you smell, how you taste, how you see, what you hear. Mm-hmm. And it's a really amazing book about the perception of any organism in the world and give you an idea of like how you can figure it out, how something else smell and see.
Wow. Well, we have whipped through a bunch of minutes here, and I could go on and on because I just find this such a fascinating topic. But thank you so much for joining us today. You have been listening to Research Matters from Cornell University. If you'd like to learn more about Christophe Duplais' research, visit the Cornell Agritech website. If you like this episode, subscribe wherever you get your podcasts and share it with a friend who loves facts as much as you do. Thanks for listening, and remember, when research meets purpose, we move closer to a healthier, fairer world. Thank you.